Good morning, dear students. Sorry for the short delay. Today's lecture is devoted to metabolism of ammonia and urea cycle. Write please the topic of lecture and we will start. At first, we need to fix the main sources of ammonia in our body. Of course, really, the highest quantity of nitrogen is coming to our body in the content of proteins. Proteins are digested in the gastrointestinal tract to amino acids. And then amino acids during processes of deamination and transamination will give us free ammonia. Also, glutamine that is synthesized in the body then will give free ammonia in kidney and liver. Small quantity of free ammonia will be generated by deamination, by degradation of biogenic amines. It's a very small quantity. Of course, our nitrogen bases during the degradation will give us small quantity of free ammonia. And finally, bacteria that are living in our intestine also can produce small quantity of free ammonia that can be absorbed in the intestinum and go to the blood. But of course, the main is dietary proteins and amino acids. Remind please yourself how nitrogen usually come and go as a free ammonia to our blood, to our body. It's formatted. Only 10% of amino acids can undergo direct deamination. We talked about this pathway and told that for this we have the special FMN or FAD containing oxidases to cut directly amino group, oxidatively cut amino group from L and D amino acids. And if for D amino acids these oxidases are working normally, but the quantity of substrates is very small, so this production is not very high. Oxidases of L amino acids, they are working very slowly due to their abnormal optimum of pH. Their optimum of pH is near 10, and due to this, they are working at 7.0 near the natural pH, very slow. So only one amino acid can be directly deaminated with high velocity, and this is glutamate, we told. This reaction is catalyzed by glutamate dehydrogenase, which is NAD or NADP containing, and this process, glutamate oxidative deamination, is strongly bound with the process of transamination of all other amino acids. So normally, flow of nitrogen, flow of free ammonia is organized through the indirect deamination of all amino acids through transamination with alpha-ketoglutarate. So any amino acid can react with alpha-ketoglutarate and as a result can be converted to keto acids. For keto acids, it is more possible to be incorporated to the catabolic pathways because no one metabolite of glycolysis or of citric acid cycle contains nitrogen. They do not contain nitrogen, so we must take this nitrogen from the amino acids if we want to incorporate them to the catabolic pathways. This process is catalyzed by a group of enzymes which are named aminotransferases or transaminases, and they are containing pyridoxal phosphate as a coenzyme. You know that precursor of this coenzyme is vitamin B6, pyridoxine. Precursor of NAD or NADP formation is vitamin PP, niacin, because nicotinamide we cannot synthesize normally in our body. Small quantity we are synthesizing from tryptophan. You told about this previous lecture, but not enough. Direct oxidases of L and D amino acids, I remind you, are FAD containing or FMN containing, and due to this, for them, vitamin B2 riboflavin is necessary. Anyway, after transamination and oxidative deamination of glutamate, we can obtain back alpha-ketoglutarate that can participate in transamination again or go to the citric acid cycle, and free ammonia. The most quantity of this ammonia will be incorporated into the urea. But about this, we will talk a little bit further. Now we need to note that ammonia, free ammonia, is very toxic product, and it can affect the central nervous system and other systems due to the changes in the formation of neurotransmitters, due to the change of pH, ATP production, and then. So let's discuss mechanisms of toxicity of ammonia, free ammonia, one by one. Write them and try to remember what things is necessary to remember to be able to explain why free ammonia is so toxic. It is soluble but toxic, and we need to excrete them by different pathways. At first, in excess of ammonia, alpha-ketoglutarate cannot be free. It will react with the free ammonia, and balance will be shifted to the formation of glutamic acid and then glutamine. At this moment, deficiency of alpha-ketoglutarate will lead to the depression of citric acid cycle. So speed of citric acid cycle will be decreased, and this will lead to energy deficiency. Then rate of transaminase action and synthesis of neurotransmitter like acetylcholine, like dopamine, which require free keto acids, also will be depleted. These two sub-reasons are combined with the decreased alpha-ketoglutarate concentration. So energy deficiency and problem in the synthesis of neurotransmitters. For example, gamma hydroxybutyric acid also cannot be synthesized in excess of free ammonia. Cause for this we need to decarboxylate glutamic acid, but glutamic acid will be significantly taken for the synthesis of glutamine. We will see this. The second point, alkalosis. The solution of 1 molar ammonia has pH near 10. So significant alkaline solutions we will obtain if free ammonia will be stored. In this alkalosis, alkali medium, unfortunately, the affinity of hemoglobin to oxygen will be increased, and this is not good, because hemoglobin with his cooperative effect must be saturated only when partial pressure of oxygen is high. It means in lungs. In peripheral tissue, where partial pressure of oxygen is lower, hemoglobin must dissociate with oxygen, and in this case, oxygen will come to the cells, peripheral cells and tissues. So in case of alkalosis that can be caused by accumulation of ammonia, hemoglobin will not give oxygen to the peripheral cells. It will be still saturated because his affinity significantly increased. It will lead to tissue hypoxia, so energy deficiency and oxygen deficiency. Third point, high concentration of glutamine in nerve tissue. At first, glutamine accumulation, as we told, decreased quantity of alpha-ketoglutarate due to the formation of glutamic acid. But glutamic acid in excess of free ammonia will be converted to glutamine, it's amide. This reaction we will discuss further. At this moment, let's fix that high concentration of glutamine will increase osmotic pressure inside of the brain and will lead to the cerebral edema. It can be dangerous, you understand, increased pressure, increased volume of cerebrum. It will cause even block of breathing or block of heart beating, and different disorders in the action of our central nervous system. From the another concentration, I told about this, now we can write that decreased concentration of glutamate will lead to the lack of gamma-aminobutyric acid, which is mediator and inhibitor of spinal neurons, normally inhibitor mediator of our central nervous system. So on the back of high concentration of free ammonia, the convulsions can be. Fourth point, ammonia ion will compete with sodium and potassium ions for the channels, for the membrane channels. You know that violation of this transmembrane transport will block the transport of many substances through the membranes, for example, transport of glucose, and also disorder conduction of nerve impulses. Because during the nerve impulses all the time the polarization and depolarization of the membranes will be done, and the special concentration of sodium and potassium necessary to keep in our cells and extracellular medium. Fifth, not last but the most important, from the most important mechanisms, it's a direct toxic action of ammonium to the respiratory center in our brain. So look, hypoxia is already observed due to the alkalosis. Energetic deficiency is already observed. Neurotransmitters are not normally produced. Inhibition, especially inhibitory action, will be not caused. And at the same moment, depression of respiratory center. So respiratory center cannot be regulated by decreased quantity of oxygen. Tissues cannot obtain normal quantity of oxygen, and this will of course increase the energetic deficiency and can cause death. So free ammonia is very toxic, and something necessary to do with this. During the evolution, different animals adapted for the excretion of nitrogen, and part of them excrete free ammonia because it is soluble. Part of them start to synthesize and excrete uric acid, which is less soluble, but if excrements are semi-fluid, it's easy to excrete crystallic substance. And finally, mammals and humans start to secrete urea, to excrete urea. So they might to develop mechanism how to synthesize this substance from the free ammonia and how to excrete it. According to the types of ammonia excretion, we can divide all of the animals to three groups. The first group is named ammoniotelic animals. Major excretory end products is free ammonia. It is possible for the animals that are living in water medium, they can excrete even through the skin, because all the time water will wet them and take ammonia from the surface of the body. Ammoniotelic animals, fishes, most of fishes, and larvae amphibia, which are also living in water. Ureotelic, excrete urea. These are humans and high animals. We are naming them ureotelic type of animals. And there is a group of uricotelic animals for which excretory product is uric acid. These are reptiles and birds. You know that birds are evolutionary from reptiles. Now this is scientific hypothesis that is known widely in scientific world. For reptiles, this was apomorphosis, and for birds, it will allow to fly they cannot take big quantity of water to excrete ammonia or urea, so the excrements are semi-fluid, and for them it is more convenient to excrete nitrogen as uric acid. As we told, ammonia is highly toxic but soluble, so it can be only excreted simply without changes only by aquatic organisms, but terrestrial organism must detoxify it first, convert to something which is not so toxic, but for example as urine, also soluble if excretion of water is the main pathway to excrete something bad from the organism, something and products of metabolism. So must be mechanism how ammonia, which is produced inside of the cell, inside of the cells of peripheral tissue, must go to liver to be converted to urea. And at the same time must be mechanisms to maintain low level of ammonia in blood. So ammonia must be incorporated into the substances which are not toxic. Simplest in our case, these are amino acids. So there are interactions between different organs of our body, between liver where urea is synthesized, between brain where ammonia must be detoxified quickly, and it will be done by the amino acid glutamine mainly. The nitrogen sources that are coming from intestinum, and also free ammonia that is coming from gastrointestinal tract produced by the microorganisms urease. Then kidney, which are doing excretion of urea and also excretion of ammonia. All of the skeletal muscles where we have formation of glutamine and alanine is the most important amino acids that will bring ammonia to the liver for the urea synthesis. Glutamine is also a substance that is important to bring nitrogen for the excretion in kidney. So the most important substances that are circulating in our blood vessels, these are ammonium, glutamine, and urea. And also a small quantity of alanine. Formation of these substances and their pathways, how to be excreted, we will discuss. And now I created more simple scheme for you, so you can write it. The most important ways how to detoxify and transfer ammonia in our organism. So in skeletal muscles, the most important pathway is the synthesis of alanine. It's a glucose-alanine cycle that we will look on the next slide. Then for our brain, this is synthesis of glutamine and asparagine. Glutamine can be also synthesized in our intestinal cells, in enterocytes, and go to the blood. Then this glutamine and asparagine and alanine, they go to the liver where they will undergo transdeamination. This scheme was the first today, and reveal free ammonia for the synthesis of urea, or incorporate the ammonia to the aspartate, because the second source for the urea synthesis will be aspartic acid. Glutamine also can go to the kidney, where it will be substrate for glutaminase to reveal free ammonia. This free ammonia is together with urea that is transported by blood from the liver to the kidney, and with ammonia salts will be excreted to the urine. So this more simple scheme, and you can write it in your notes to understand the way of ammonia and to fix the name of most important substances that we will discuss today. About alanine formation in the skeletal muscles. In the period between meals, muscle proteins can be degraded to amino acids. These amino acids all can give their amino group to pyruvate, because pyruvate is formed in skeletal muscles, you know, normally during the anaerobic part of oxidation of glucose in muscles. Lactate is formed, but after oxygen will come, after normal vascularization of the muscles and increase of volume of inspired oxygen, pyruvate is reconstructed, and here it can react with the any amino acids that before reacted with alpha ketoglutarate and put their nitrogen to glutamic acid, and simply be converted to alanine. Alanine is a small water-soluble non-toxic amino acid that can go by the blood to liver, and they react with alpha ketoglutarate to form glutamate, which after action of glutamate dehydrogenase will give urea, ammonia for urea synthesis, because urea cycle localized only in liver. Muscles cannot neutralize free ammonia themselves. So alanine is the most important amino acid that is formed in muscles. Glutamine is also formed, but in less quantity, if we will talk about muscles. Pyruvate, that will be revealed after this, can be used in liver for gluconeogenesis, and glucose can return to the muscles by blood. It's very similar to Cori cycle that we learned in the first semester, but Cori cycle, this is glucose lactate cycle. It means lactate after formation, if it cannot be converted back to pyruvate, it means if oxygen is not coming in enough quantity, lactate can also, same like alanine, swim to the liver for gluconeogenesis. So here you can see the same transport of carbon that is formed from glucose, it means 3-carbon substance, pyruvate, that is formed from glucose during the degradation of glycogen and glucose, glycogenolysis and glycolysis in muscles, and relationships with gluconeogenesis. But in case of alanine cycle, we also organize transport of nitrogen, of amino groups, from muscles to liver. And this is very important because, as I told already, urea can be synthesized in our body only in liver. It's a big mistake to tell that urea is synthesized in kidney or somewhere else. There is no active erginase in any cells except of liver. Next important reactions, these are reactions of formation of glutamine, amide of glutamic acid. Ammonia can be fixed by alpha ketoglutarate. It is written as a free, but really, usually glutamate dehydrogenase catalyze degradation of glutamic acid with participation of NAD. Back reaction, glutamate dehydrogenase, it's a reversible reaction, but in back direction, usually coenzyme is NADP. We are obtaining at first glutamate, glutamic acid, which after this will react with the ammonia and amide of glutamic acid. Glutamine is formed. Look that for this reaction, it's a synthetic reaction, ATP is necessary. This glutamine can be transported to liver, where it will be cut by the action of liver glutaminase. It is present in liver mitochondria. And after this, free ammonia can go to the urea cycle. for the synthesis of ammonia. The glutamate that is revealed from the glutamine, after this can be deaminated, oxidatively deaminated by glutamate dehydrogenase and returned to alpha-ketoglutarate. Alpha-ketoglutarate can be used for transamination, for the new glutamine synthesis somewhere, or can go to the citric acid cycle to obtain energy or to synthesize glucose finally from the products of Krebs cycle. You know, they are all converted to oxaloacetate, which is initial for the synthesis of glucose. From another point of view, glutamine can also by blood transported to the kidney, where it will be substrate for kidney glutaminase, and this is renal ammonia genesis, then to glutamate, then by glutamate dehydrogenase ammonia can be by glutaminase ammonia can be revealed. Glutamate dehydrogenase, after action of oxidative deamination of glutamate, new portion of ammonia can be released. So 2 amino groups we can obtain from glutamine. And alpha-ketoglutarate in kidney also can be used for the gluconeogenesis. You know that near 50% of gluconeogenetic pathway is localized in kidney. They also a little bit glucose synthesized organs. And or can be cut till obtain final products of metabolism. Free ammonia can be excreted by kidney to urine. It's about glutamine degradation. From glutamine, free ammonia, if it comes to kidney, if glutamine comes to kidney, free ammonia is excreted and also used for the regulation of acid-base balance of whole organism. It's important, so about this we will talk a little bit more detailable. So these reactions, please write. Formation of glutamate from alpha-ketoglutarate, then glutamine synthetase reaction with ATP formation, transport to mitochondria, transport to kidney, where glutaminase is acted. Glutaminase reveals free ammonia. In liver, it can be used for urea synthesis immediately. Urea after this will be excreted by kidney. In kidney, directly free ammonia is released and excreted, used for the maintenance of acid-base balance, regulation of acid-base balance. So about participation of nitrogen, of ammonia in the maintenance of pH of our blood and extracellular medium. Action of two enzymes, glutaminase and glutamate dehydrogenase. In the tubular lumen, ammonia ion, NH4+, is produced from ammonia and hydrogen. So this equation, ammonia plus hydrogen, ammonium ion. This reaction is favorite the acid pH of urine. Deformed NH4+ in the tubular lumen cannot easily cross the cell membranes and travel to the lumen to be excreted in urine with other anions such as phosphate, chloride, as a salt, like a salts of ammonia, ionized. Forming ammonium phosphate, ammonium chloride, ammonium sulfates. So this production of NH4+ in the tubular lumen account about 60% excretion of hydrogen ions associated with non-volatile acids. So acidic hydrogen is excreted together with ammonia, and this helps to kidney to maintain acid-base balance. So this is the example where the bad thing, ammonia, we are using for very important function to maintain the acid our pH, our acid-base balance. And this is very good adaptation because ammonia anyway must be excreted, but also it is used to excrete excess of hydrogen ions from our body. One more reaction is necessary to write. This is synthesis of asparagine. It can be synthesized from aspartic acid and glutamine. In this case, enzyme asparagine synthase will incorporate this nitrogen instead of carboxylic group. Second radical carboxylic group will be changed to amide group, and we will obtain asparagine and glutamate that can participate in the reaction of synthesis of glutamine again. Why? Because it's another substance and it also can help to transport nitrogen in the inside of the structure of asparagine, because asparagine water-soluble, non-toxic amino acid. But what is important, what necessary to say? Of course, asparagine synthase also can work when instead of glutamine we will take free ammonia. So free ammonia also can be incorporated to the aspartate by asparagine synthase. Problem is that a little bit more energy is necessary for this process. Look, if for glutamine synthesis 1 macroalgae bond we cut it and ATP was converted to ADP, here 2 macroalgae bonds we are losing for synthesis of asparagine. Due to this, lower role belongs to asparagine as a transport, and the most important for us is of course formation of glutamine. Glutamine is formed in nerve system, as we told, in enterocytes, a little bit in muscles, where biggest quantity of ammonia will be taken in the structure of alanine. And it's easy to understand why glutamine, glutamic acid, alanine are the most widely distributed amino acids in our blood, because they help us to detoxify and to transport ammonia to kidney for excretion and to liver for their urea synthesis. Okay, before we will start to talk about urea cycle, urea synthesis, we will do the small break, 10 minutes, and then continue. Okay, let's return and continue our lecture. The second part will be devoted to the process of urea synthesis. Let me introduce you the molecule of urea. Who knows the formula of acetone dimethyl ketone? We have CH3 here on the side. Should note that these molecules are similar, but urea is a diamina ketone. Two amino groups are present and combined with the keta group. This is the ball model of this molecule, and we need to write two important things about this molecule. At first, that it is formed in liver but excreted from the organism together with urine. It is not formed in kidney, and liver is not doesn't produce urine. Urine is produced by kidney, but synthesis of urea, formation of this molecule, will be done in liver and only in liver in our body. Part of enzymes from the urea cycles present in other tissue, but not all. So only liver, only hepatocytes can produce urea. If they will die, for example, as a result of liver damage, liver diseases, ammonia will be accumulated in the body. About this ammoniemia we will talk a little bit later. At this moment, write please important things, that normal blood content of urea is from 3.3 to 8.3 millimole per liter. Put attention that the concentration of urea can be higher in our blood than concentration of glucose. So it's a normal metabolite, but the concentration must not be higher than 8.3 millimole per liter. Sometimes, due to the high rich protein diet, it can be a little bit higher than 8.3, but usually if patient is in health, it will be not higher than 10 millimole per liter. So if it is really higher than 10 millimole per liter, you must suggest the presence of disease in the patient and find the reason for this uremia. Discovery of ornithine or urea cycle, it's a Nobel-Prize discovery of Hans Krebs and his student Kurt Henseleit. In 1932, they proposed to the scientific community the scheme of urea cycle, ornithine cycle, and the third name is the Krebs-Henseleit cycle. It's necessary to note that they discovered it's earlier than real Krebs cycle, citric acid cycle that we are naming Krebs cycle. So according to the historical direction, ornithine cycle was a Krebs cycle number 1, and TCA, citric acid cycle or tricarboxylic acid cycle, is cycle of Krebs number 2. So at the moment when energy production sequence of acids was not known, Krebs and Henseleit suggest cyclic sequence of reactions which helps to organisms synthesize urea from free ammonia. So let's discuss the reactions of Krebs cycle. Not so many like in TCA, but we need to discuss each reaction, name each enzyme, and look what will be done according to the mechanisms, what substance will be formed, how these reactions are regulated. So the first step, it's a synthetic reaction where carbon dioxide is condensating with free ammonia. So the first source of nitrogen in the urea formula is free ammonia. For the formation of necessary for our substance, 2 ATP molecules are necessary and water is necessary. This reaction can be done only in mitochondria and mitochondria of liver, of course, mitochondria of hepatocytes, and substance that is formed is named carbamoyl phosphate. Carbamoyl, this is a residue, look, combined carbon dioxide and amino group. Phosphate will be taken, as you understand, from ATP, and this macroenergic substance is named carbamoyl phosphate. Look how many energy is necessary for the production of 1 molecular carbamoyl phosphate. We are using 2 ATP and obtain 2 ADP. It means the 2 macroenergic bond we are using for this. So it's not cheap process. We need energy, and this one of the linkage to the Krebs cycle, which is ATP producing sequence of reactions. Note, please, write the reaction, show where nitrogen is incorporated, where carbon dioxide is incorporated, show the 2 macroenergic bonds we are using for this reaction, that reactions will flow in mitochondria of hepatocytes, and name, please, enzyme. Enzyme easy to create because as for glycogen synthesis, as for glutamine synthesis, as for ATP synthesis, we are adding synthase to the name of the synthesized product. Carbamoyl phosphate synthase. Carbamoyl phosphate synthase, it's the name of enzyme. And note, please, that this is the more slow reaction in all of the sequence, and as usually slow reactions are regulative reactions. Carbamoyl phosphate synthase is regulated by the substance which is named N-acetyl glutamate. It's a residue of acetate that is combined to glutamic acid, and so glutamate not only participant in the metabolism of amino acids, but also one of the regulator for urea synthesis. Allosteric activator of carbamoyl phosphate synthase is N-acetyl glutamate. Structure is not necessary to learn, but it's easy to imagine it. It's acetylated glutamate through the amino group. In its order, N-acetyl glutamate, it's a result of reaction between glutamic acid that is coming with proteins, with whole pool of our dietary amino acids, and who is the carrier of residue of acetate? Of course, acetyl-CoA. So acetyl-CoA is produced in great quantity when diet contains normal quantity of carbohydrates and supply us with enough quantity of acetyl-CoA, even maybe with excess, which can be converted to fats. Anyway, understandable that directly regulating influence to the synthesis of urea will be caused by quantity of proteins. Due to this, I told you that in very rich with proteins diet, quantity of urea that can be produced by liver can be higher than 8 millimole per liter. Right place reaction indicate that N-acetyl glutamate is the activator of carbamoyl phosphate synthase, main regulative reaction for all urea production. And let's go to the step 2. In step 2, carbamoyl phosphate condensates with amino acid ornithine to produce citrulline. Also, this reaction still occurs in mitochondria of hepatocytes and catalyzed by enzyme which is named ornithine transcarbamylase. We transfer this group, look, to ornithine with synthesis of citrulline. Phosphate will go free and macroenergic bond that was here in carbamoyl phosphate is used for this synthetic reaction. Look to the structure of these acids, ornithine and citrulline. It's interesting. Actually, they are amino acids. They both contain carboxylic group and amino group. Moreover, they are alpha L amino acids. Look, amino group is combined to the alpha atom here. Amino group is combined to the alpha atom here. So both ornithine and citrulline are alpha L amino acids, same like all 20 proteinogenic amino acids. And I like to ask my students the question, why in this case we did not study ornithine and citrulline between 20 proteinogenic amino acids? Answer is that they are not be used for the synthesis of natural proteins. Actually, we have more than 300 different amino acids in our world. Near 40 of them exist inside of our body with special functions. But not all amino acids are used for the synthesis of protein. Moreover, not all alpha L amino acids are used. You know the D amino acids are not used, beta, gamma, delta amino acids are not used. But here we have very similar to proteinogenic alpha L amino acids, and anyway they cannot be found in the structure of proteins. Why? Because appearance of amino acids in the sequences of polypeptide chains is determined by our genes. The sequence of amino acids is written in our genes by triplets of nucleotides. So we have genetic code. We have a correspondence between triplets of our nucleotides with different 4 type of nitrogen bases and our 20 proteinogenic amino acids. So for citrulline and for ornithine we have no codons, we have no triplets of nucleotides, and due to this they never will be incorporated into the structure of polypeptide chains, into the structure of proteins. But they perform very important function. They participate in the formation of urea. So let's write this reaction. Ornithine transcarbamoylase reaction via carbamoyl phosphate condensates with the ornithine. And due to the reconstruction of ornithine at the last step of urea cycle, the synonym name for urea cycle is ornithine cycle. Ornithine, you can find this root ornithus, yes, in the words that are devoted to the birds. Ornithologists, these are specialists who study birds. Ornithus, ornithosis, it's a disease which is combined with the birds. Actually, ornithine was isolated from the primary was isolated from the feces of the birds and from the feather, bird feather. Due to this was named such a ornithine. Look to these substances, also alpha L amino acids, nothing was changed here, only additional group was added, this group that is colorated in blue. Citrulline, not from citrus, not from lemon. Citrulus in Latin it's a watermelon, and due to the isolation of these amino acids from the watermelon it was named citrulline. Step 3, a little bit more complicated substance is formed, but really if you will subsequently create formula of citrulline, formula of aspartate you know very good, because it is negatively charged amino acids, only 1 from 2 of them, and usually glutamate and aspartate structure is very well known by students. In this case, easy to create the substance that is formated on the 3rd step of urea cycle. Citrulline is transported to cytosol. At this moment, reactions will flow in cytosol. It's also important to remember that this cycle, in comparison, Krebs cycle number 2, TCA, is localized completely inside of mitochondria. Here we can see how in the middle of the cycle, substances are transported to the cytosol and reactions will be continued in cytosol. New reaction of condensation. Now citrulline will condensate with aspartate, and look, the second nitrogen to the urea will be brought by aspartate. It's important to remember because aspartate can be formed during the reaction of transamination with oxaloacetate of any amino acid. Aspartate can be formed by reaction oxaloacetate. You know, this is a metabolite that present in all of our cells because it's a part of our citric acid cycle, and this is the initial substance for gluconeogenesis. Not in high quantity, but oxaloacetate is present in our body. Aspartate can be taken from the dietary proteins also, and this is the direct way how to take nitrogen from the aspartate. Again, ATP. ATP is used for the condensation reaction, and again, note that we are using 2 macroergic bond here because 1 molecule of ATP is converted to molecule of AMP, not ADP. Aspartate is colorated, and you can see how through this amino group our aspartate combined to the citrulline, and this in this structure, the arginine can be recognized if we will surround by this way. Look, this dicarboxylic saturated acid from 4 atoms of carbon, and you also know good this substance because this is succinate, also metabolite of citric acid cycle. So the name of substance will be argininosuccinate. As a result of aspartate and citrulline condensation, we obtained argininosuccinate, new substance which is half of arginine and half of constructed from succinic acid. So what is necessary to write about this reaction? That argininosuccinate synthase is an enzyme, so also creation of names of enzymes for urea cycle is not very difficult. And of course, it is necessary to remember that here next step of usage of ATP. As I told you, urea synthesis not cheap process. We need to use ATP for carboxyl phosphate production, and here for the condensation of argininosuccinate, and for production of argininosuccinate also ATP is necessary. At the next step, finally cleavage. Condensation, 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 finally cleavage. Argininosuccinate cleave to produce the amino acid arginine and non-saturated acid with 2 carboxylic group, which is also very good known. It's a fumarate, which is metabolite of citric acid cycle. Cleavage, so the name of enzyme will be not synthase, of course. This is argininosuccinate lyase. Lyase. And it will cut here. So bond hydrogen will be taken to fix amino group here, yes. And due to this, the double bond will be formed in the structure of former aspartate, succinate. Here we will see fumarate, transisomer, it's a fumarate. And amino acid arginine. This formula you also very good known. So it is not easy to write all the reactions of urea cycle with formulas. I hope that all will be able to do this during the control point and during the exam, because we are working with the known for us substance metabolites of TCA or proteinogenic or very similar with proteinogenic amino acids. Step 5, final step, revealing of urea. Final reaction will be catalyzed by the enzyme arginase, and this is limiting of presence of this reaction only in hepatocytes. As I told you, arginase is a gene of arginase is expressed only in hepatocytes, and only there we have such a enzyme for the cleavage of arginine to urea and back to ornithine. Look to the mechanism of reaction. Our urea is cleaved here. So 2 nitrogens, one from free ammonia, one from aspartate, will come to the structure of urea. What is necessary for this? Enzyme is orgeanase, and ornithine is reconstructed. Let's write this reaction. Same like oxaloacetate is reconstructed in the citric acid cycle. Here ornithine is also will be reconstructed at the last reaction. Urea after this will be excreted from the hepatocytes to blood, swim by the blood to kidney, where it will be filtrated to the primary urine, then concentrated and excreted with the secondary urine out from the organism. So this is overview, and now you can see that it is really cycle. Yes, carbamoyl phosphate is formed in mitochondria and condensate with ornithine. Citrulline is formed, transported to the cytosol, then converted to arginine succinate, reacting with aspartate, then will be cleaved with formation of fumarate and arginine, and then final step, arginase will cut urea, free urea, and reconstruct ornithine. Ornithine back will be transported to the mitochondria for the reaction with the new molecules of carbamoyl phosphate. The most important regulative enzyme, this is carbamoyl phosphate synthetase, which is regulated by N-acetyl glutamate. One more attention to the two sources of nitrogen atoms in the urea. Two different substances, free ammonia and aspartate. So this is one of the important role of aspartate as a dicarboxylic amino acid in the formation of urea. Many times we repeat that this is metabolite of the citric acid cycle. This is a metabolite of citric acid cycle. Really, our cycles, urea cycle and TCA cycle, are combined one to another. Look, aspartate that is formed from oxaloacetate by the reaction of transamination will bring second nitrogen for the urea formation. Fumarate that is formed after this can be converted to malate, and both fumarate and malate is a combines the urea cycle with citric acid cycle. So there is a stable term, Krebs bicycle, because two cycles combined with the several pathways, and they depends one to another, not only by this substance but also by ATP production. So look, for the synthesis of urea, ATP is used, many ATP. It's 3 molecules and 4 macroalgic bonds of ATP necessary to synthesize 1 molecule of urea. So the portrait of our famous Hans Krebs on the back of its citric acid cycle, and remember about this bicycle of Krebs. Both cycles are not isolated. Now we need to fix our attention on the disorder of metabolism of ammonia, and for this please write the definition of the important things about important term that we are using to describe normal ammonia metabolism and the disorders of it. Residual nitrogen. Write please that this is all nitrogen of non-protein blood compounds, nitrogen of substances remaining after removal of blood plasma proteins, because of course greatest quantity of nitrogen is contained by our blood serum proteins, albumin, globulins, immunoglobulins. So if we will take all of these proteins, all residual remaining nitrogen compounds will be represented by urea, near 50%, amino acid nitrogen, because free amino acids, we told, are swimming can be absorbed in the intestine and can help us to transport nitrogen to prevent intoxication with free ammonia. It's near 25%. Then uric acid, the product of urine metabolism, degradation of urine nitrogen bases. Creatine and creatinine compounds, nitrogen-containing compounds combined with their muscles work. Ammonia, free ammonia, and indican look that not more than 0.5% from all nitrogen-containing compounds. And small, less than 0.5% compound, which are also, don't forget that they are nitrogen-containing but not protein. Glutathione, bilirubin, and others. Minor components. The most important, of course, urea, more than 50% of residual nitrogen. Right place the normal content of residual nitrogen in the blood. It is the oriented for the decision will be metabolism of ammonia and urea done correctly or not. This is 14.3 to 25.0 millimole per liter. Please put the attention that in our country we are using C units to represent important values of different biochemical components of blood and urine. Residual nitrogen 14.3 till 25.0 millimole per liter. Increased residual nitrogen in the blood can be due to the increased quantity of different compounds from the previous list, but totally increased quantity of nitrogen containing compounds in the blood can be named by Latin term azotemia. Azot, it's in Latin nitrogen. So high concentration of nitrogen containing compounds in the blood, except of protein, residual nitrogen we are discussing, can be due to increased free ammonia. In this case, it is named ammoniemia or hyperammoniemia, or increased quantity of urea. In this case, this type of azotemia will be named uremia. Put attention that hyperuremia we are not talking. Hyperammoniemia sometimes is used, but hyperuremia no. Uremia, it means urea higher than norm. Ammoniemia and hyperammoniemia means ammonia higher than norm. Put attention that absolutely two different reasons leads for the uremia and ammoniemia. Ammoniemia is resulting of absence of urea formation, and this can be due to the lack of enzymes of urea cycles, enzymopathias, or liver damage, liver insufficiency, when our factory, only one factory can synthesize urea in our body, this is liver, so liver failure will be represented, will be manifested as ammoniemia. Uremia, usually due to the hyperproduction of free ammonia and hyperproduction of urea by liver, in this case as an adaptation process, or due to the renal failure when excretion of urea is disordered. Let's talk about this a little bit more detailedly. So liver damage or metabolic disorders associated with the elevated ammonia can lead to such symptoms like tremor, slurred speech, blurred vision, coma, and even death. We divide in hyperammonemia according to the reasons to the acquired, due to the liver disease, usually chronic hepatitis, cirrhosis of liver, or obstruction, biliary obstruction and inhibition by bile, our hepatocytes where they cannot work, cannot synthesize urea. And hereditary hyperammonemia due to the enzymopathy, due to the genetic deficiency of enzymes of urea cycle. Actually, each of 5 enzymes described as a deficient, and different types of ammoniemia with different difficulty of this can be in patients with hyperammonemia. More detailable symptoms of hyperammonemia are represented here. Instead, in addition to tremor, as I told you, seizures can be due to the neurotransmitter lack, like gamma-aminobutyric acid. Then enlargement of liver may be combined with the liver disease. If it is chronic, growth retardation will be, hypothermia can be observed, and if ammonemia is developing as acute situation, of course the disorders mainly will be from the neurological problem. It's a tremor, seizures, ataxia, and slurred speech, disorder of vision. So in acute situation, the primary symptoms will be from the central nervous system. But it can be chronic at the lower level. In this case, more possible, this is one of the type of enzymopathy. Here I represented for you possible situation, possible urea cycle disorders in dependence from what enzyme is deficient. Note please that inherited hyperammonemia usually without metabolic acidosis will be more characteristic respiratory alkalosis, so hyperventilation and respiratory alkalosis. This is ornithine transcarbamylase deficiency, carbamyl phosphate synthase deficiency, citrullinemia, argininosuccinic acidemia, or argininemia. It depends from the points of the deficient enzymes. We know and learn already that in enzymopathies usually, if enzyme is in lack, substrate of the reaction will be accumulated, product will be in low concentration. We are not learning detailable of them. You must only know that all 5 enzymes can be deficient, and this will lead to the inherited ammonemia. Usually, of course, it will develop after birth. Kidney damage or metabolic disorders which are combined with elevation of urea are manifested by involuntary muscles twitching, progressive weakness and easy fatigue, loss of appetite due to the nausea and vomiting, diarrhea can be observed. In chronic situation, this is muscle atrophy and abnormal mental functions also. But here, if a situation is not acute and urea will start to accumulate slowly, on the first plan will be symptoms from the gastrointestinal tract. It's nausea, vomiting, poor appetite, and only then more slow symptoms will develop from nerve system twitching, muscle atrophy, dryness of skin, and then swelling, which will accompany diseases of kidney if there is a retention of fluids also. So here we also can talk about 2 reasons for the uremia. It's a production uremia, also called prerenal, when increased production of urea is not combined with the liver, with the kidney disease. Liver is overproducing urea a little bit more, that because it can increase velocity of production of urea several times. It can be in high protein diet, I told about this, increased protein catabolism. What reasons? Stress, fever, major illness, for example, degradation of tumors, or in leukemia and other malignant tumors of blood cells. Corticosteroids therapy is also. So when cells are degrading, of course, great quantity of proteins are degrading, and overproduction of the free amino acids will lead to the overproduction of urea. The second type, this is a retentional uremia. It can be renal or postrenal. It depends from the reasons. Here, urea is accumulating due to the kidney insufficiency. to excrete it, to filtrate it. It can be in chronic kidney failure, acute and chronic glomerulonephritis, it means filtration is disorder, or tubular necrosis where concentration is not possible, and other kidney disease, and also due to the urinary outflow obstruction, such like in kidney stone disease or tumors, or tumor of urinary bladder or prostate, or severe infection where it is blocked by pus and the cells which are desquaming from the epithelium. For this topic, I will tell you, thank for the attention, but this is not the finish of the lecture, because we need to tell a little bit about next lecture. Next lecture also will be mine, and we need to discuss metabolism of complex protein. So let's use our time for looking and analyzing of several previous slides about complex protein. I need to remind you what is the difference between simple and complex proteins. So please write the topic of the next lecture, and let's remind classification of our proteins to simple and complex. We have a little bit more time for this. Let's remind that if proteins are constructed only from amino acids, so after hydrolysis, total hydrolysis of the protein, we can obtain only amino acids. It does not matter how many polypeptide chains was in protein, one or more. If only amino acids, protein will be simple. We will name it simple. Opposite group, these are complex proteins, in which we have protein part, which is constructed from amino acids, and non-protein part. It means something non-protein. It can be phosphate group, it can be carbohydrate, nucleotide, it can be chromo group, lipids, it can be metal. And according to this non-protein part, we are separating all complex proteins with two: phosphoprotein, glycan, nuclear protein, lipoproteins, and then the most important groups are represented here. The problem is that in part of the textbooks, complex proteins are named conjugated proteins. And for the complex means quaternary structure of proteins. It means complex, these are more than one polypeptide chain. Please, my dears, let's make the agreement that if protein contains more than one polypeptide chain, we are talking about quaternary structure. But if all these polypeptide chains constructed only from amino acids, anyway, this quaternary structured protein will be simple. And we will use term complex protein instead of conjugated protein for the proteins that can be divided to protein and non-protein part. Examples of simple proteins: albumin, growth hormone, they contain only amino acids. Complex proteins: dehydrogenases, which is NAD and FAD as a non-protein part. Our chromoproteins, like hemoproteins, metalloproteins, like transferrin, which are necessary to transport iron. Most of our proteins, of course, are complex because they are working together with their non-protein parts. For example, complex enzymes are not working without their non-protein parts, without their coenzymes. But there are simple enzymes like pepsin, lipase, amylase. They do not require something non-protein for their action. So please, my dears, we will start metabolism of complex proteins on the example of hemoproteins, proteins that contain heme, and nuclear proteins, because with the disorder of this, metabolisms are very severe complications like jaundices in case of heme disorder of catabolism, and GOAT in case of nucleotides disorder, nucleotides catabolism disorder. So about these two groups we will talk, and we will start from hemoproteins. So let's look to the structure of heme. This is the ball model. We have 4 heterocycles from 5 participants. One of them is nitrogen. Nitrogen, 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 nitrogen. Pyrrole rings, so called here. Inside atom of iron with coordinate number 6. So 4 bonds for nitrogen, 2 covalent, 2 coordinate, and 2 bonds, 1 for oxygen and 1 for residue of histidine from the protein part. Each subunit of hemoglobin, there are 4 subunits in hemoglobin, combined with this structure. Look that porphyric rings also have the small antenna radicals that are looking out from the plane of the total heme. These are residues of propionic acid, these are oxygen, and these are methyl radicals and vinyl radicals. Their positions depends from the numeration of pyrrole rings. And this is the plain formula. Once more, here we have iron, vinyl, methyl, methyl, propionic, propionyl, methyl, vinyl, methyl as the radicals, and 4 pyrrole rings surrounding atoms of iron. And here you can see how this heme incorporated into the polypeptide subunit. Main structure is alpha helix in the subunits of hemoglobin, and inside to the residue of histidine our heme is combined. Hemoglobin contains 4 such subunits in comparison with myoglobin, which contains only 1. This comparison between myoglobin and hemoglobin we will do in lecture about biochemistry of blood. At this moment we will finish, and next lecture will be devoted how this molecule is degrading. Actually, we can synthesize heme and other porphyric rings in our body, but cannot cleave them completely till CO2 and water. And due to this, we need to learn the special pathway that heme will undergo till excretion from the body. Thank you for the attention. Thanks to the small quantity of students that were with me together exactly at this moment during the translation. I hope that you were in time to conspect everything, and wait you for the next lecture next week for the discussion of catabolism of complex proteins. Good luck!